1 year on, what have I done? Well, just to review basic cell processes, which is helpful. Yeah, we inherit our DNA from our parents, and this is unchanging throughout our lives, or virtually unchanging. Day to day, as our cells undergo different activities, this is transcribed into messenger RNA, and this then is translated into protein, which performs the various functions that the cell requires. So, utility of these studies of genomics and proteomics can be divided into studies of inheritance or inherited likelihood of developing a disease, pathogenesis, or how that disease comes about, how it's mediated, the disease mechanisms, and also studies to aid our diagnosis. So the inheritance, we can identify markers in the DNA of chronic fatigue syndrome. And these are various types, the simplest of which is the single nucleotide polymorphism. And these have possible relevance to pathogenesis and are a possible route to treatment, but probably unlikely. Pathogenesis then is probably best studied through RNA expression and protein expression. And through understanding this, we can design new treatments. We can aid in the diagnosis of a disease through study of its RNA expression and protein biomarkers. So the molecules of interest then are DNA, RNA, and proteins primarily. So we have studied principally the messenger RNA expression in the peripheral blood of patients with chronic fatigue syndrome and compared this with normal subjects which we've obtained from the National Blood Service. Our cases of chronic fatigue syndrome fulfill the CDC criteria, and most of these also fulfill the Canadian criteria. And these were taken from a CFS clinic in Dorset primarily. The cases were age-matched with normal people from the National Blood Service, 1 case to 2 controls. And we performed microarrays on these patients. We took a blood sample, extracted the total RNA, and did the microarray experiments using the Affymetrix U133 plus 2, which represents all known messenger RNA transcripts. And we analyzed these thousands of gene levels, which are converted from these colored spots into numerical values using GeneSpring, and compared all the patients with all the controls to find those genes which were expressed at much higher or much lower levels than the normal people. So then we have a gene list that we need to confirm then using a second method. The first method looks at all the genes at once. The second method looks at one gene at a time, and in this case we used TACMAN PCR. In this we look at a single test gene and compare that with a normal housekeeping gene of the cell, which we know is expressed at the same level in all the patients and all the controls. And it's this ratio that gives us our results. And there have been various studies using this type of approach. Some don't use TACMAN, and those are the ones from the CDC, unfortunately. But generally, all of these implicate immunity and defense as being the number one abnormality. And this fits with our epidemiology studies in which we have implicated a range of different virus infections and ongoing immune abnormalities. But we don't yet know the details of that, and so we took this larger study. We set out by screening samples using the microarray, as I've described, and this other screening method, and these work differently. This is massive parallel signature sequencing, which basically sequences all the total RNA, all the messenger RNA molecules in the sample, and provides a signature sequence for each with an absolute copy number for each of those. This is a relative method. This is an absolute method. This method is capable of detecting new sequences. This is only capable of detecting sequences that you have prepared probes for on the array. We thought initially this would help us with the human genes in that we might find interesting translocations, new genes, etc. It didn't. We didn't find anything new out of this study for the human genes. So we have used this for a virus study, which I'll tell you about in a moment. So back to the microarray then. We have confirmed now 80 human genes which are associated with chronic fatigue syndrome, and 78 of those are upregulated and 2 are downregulated. And again, we have the immune response. Also, genes involved in neurological function, mitochondrial genes, selective regulation, G proteins. Other features are transcriptional regulators, translational regulators, etc. And this is a slide showing just a few of these. On this axis, we have the mean relative quantity, and that is basically the normalized level of each test gene in all the patients, shown in the cross-hatched, and all the controls, the normals, shown in black. And you can see that all these genes have higher levels in the cases, the CFS, than in the controls. And we published a pilot study 2 years ago where we described 16 genes were abnormal in chronic fatigue syndrome. Well, I can tell you that 15 of those have confirmed in the same direction as we found in the previous study, and 1 did not confirm. The interleukin-10 receptor alpha, which we previously was the only down-regulated gene in that study, we found to be significantly upregulated this time, which was the only gene that didn't agree with the pilot study. We have a large number of these eukaryotic translation initiation factors, EIF, which are some of them linked with virus infection like this EIF4G1, others linked with neurological disease like this 2B4, but all of them are important for production of proteins and are linked with mitochondrial function. So this, we've analysed the genes in various ways, and it's deliberately blurred. I apologise for that. It's because I don't want anybody to take down the gene names at this stage before we've published that, and the reason for that is a good one, in that it may take us a year or more to publish this paper because people don't like taking papers in chronic fatigue syndrome. But anyway, we've analysed the genes in various ways, and this is an analysis in Ingenuity Pathways Analysis, which is a sophisticated program used by drug companies to analyse their gene expression and other proteomic data. And there are various networks that these genes can be organised into, and basically there are 4 distinct networks which can all be connected together as shown here. And these are clustered according to the cell compartments. This is the nucleus. This is the cytoplasm, this is the periplasmic space, and this is the plasma membrane up here. The genes that we find, the 80 genes, are shown here, either red or blue, and the red ones are upregulated and the blue ones are downregulated. So we only have 2 that are downregulated. The rest are upregulated to various degrees. So we have various transcriptional regulators here in the nucleus. Translational regulation, the eukaryotic translation initiation factors here. Some cytokine molecules up at the top here. And certain features of this cell signature suggest virus infections. And so we're testing for a number of things to do with herpes virus infection before we can publish that. But it shouldn't take us too long to do that. We analysed the genes for overrepresentation of transcription factor binding sites. Now what that means is every gene has its main sequence. But prior to that sequence, there's a little regulatory region which has binding sites for so-called transcription factors. And these, for example, Jun, Phos, SP1, shown here, bind this region, and they either increase the transcription or they decrease the transcription. And what we look for in our 80 genes is the footprints of these transcription factors, the places that they can bind, to see do we have any particular overarching hypotheses in terms of does 1 or 3 transcription factors, may that account for the majority of our signature, for example. And we found that 12 were abnormal. And one of these is a virus-linked transcription factor. So we're now testing for those in our samples along with the virus studies. So next steps are we need to test other disease controls, more patients, more normal people, and disease controls such as patients with other infectious diseases and other rheumatic diseases, for example. Depression is an important disease control group. So that we can say if we find these gene abnormalities in these other diseases, then they're not specific to chronic fatigue syndrome. And we can then take those out and reduce our gene signature. We need to look at the variation with time of these genes, and we're going to look at a small subset of patients at monthly intervals over a year and see which genes are, what the variability is, and within individual patients, do they maintain the same signature or does that vary? And maybe link that with symptoms as well. And then we need to look and see of the genes that we're left with after that, which gene abnormalities occur in the different white blood cell subsets. Because so far we've only looked at all the white blood cells in the peripheral blood as a group. There are, as we know, various types of lymphocytes, T cells, B cells, there are monocytes, there are natural killer cells, and we need to know which gene abnormalities occur in which cell types. And we're also looking at microRNAs, which are a new, relatively recently recognized class of RNA. These are 22 nucleotides long, compared with several hundred to several thousand nucleotides for the messenger RNA species. And they basically degrade or sequester messenger RNA species. So it is possible that a messenger RNA gene signature that I've just described could be accounted for by abnormalities of microRNAs, and even relatively few, because one microRNA can target several hundred to several thousand messenger RNAs, and so that's a possibility. Another exciting possibility of study of microRNAs is that they themselves can be used as treatments. So we have done a microarray study comparing 15 chronic fatigue syndrome patients and 30 normal people, matched 2 to 1 again, and we found that there are approximately 40 microRNAs that are differentially expressed between those groups. And we're now confirming that by testing them individually by TaqMan PCR. So there are various protein abnormalities, as we've heard mentioned before by Dan, in chronic fatigue syndrome. And most recently, and very excitingly, the Japanese have reported a visual near-infrared spectroscopy study which efficiently differentiated chronic fatigue syndrome patients from normal people. And this was published last year and presented in Fort Lauderdale in January. And basically this provides, along with the study by Baraniuk, which found a CFS-related protein signature in chronic fatigue syndrome, provides a rationale for looking at proteins in this disease. Incidentally, one of these proteins found in the CSF was gelsolin, which is one of the genes we published in our pilot study, which we have confirmed is abnormally upregulated in chronic fatigue syndrome in peripheral blood. So our approach to development of a diagnostic test is to look at the serum samples and the proteins in that using surface-enhanced laser desorption and ionization protein chip, which is a fancy method of mass spectrometry. And basically, fractionated serum samples from the chronic fatigue syndrome group and the normal group are spotted onto these protein chips. These are then excited by a laser, and the ions coming off that travel down a column, and their time of flight is detected and reproduced in a mass spectrum like this. We can see then that this illustrates the principle of the method, where we can, the CFS people are illustrated in red and the normal people in black, and the arrows refer to peaks that are different between the two groups, and this is the basis of the method. So we've done this now in about 40 chronic fatigue syndrome patients and 40 normal people, looking at fractionated sera, 6 fractions from each subject, in 3 different chip surfaces for each fraction. And we've identified 15 protein biomarkers that are statistically significantly different between the two groups. And these, basically, this is an entirely statistical study. We could be looking at virus proteins, we could be looking at human proteins, we don't yet know for sure. But these are very different between the two groups. We're currently identifying these using a combination of 2-dimensional gel electrophoresis and mass spectrometry. We're going further to look at more patients, more normal people, and the same disease controls that we'll be looking at for the gene studies. Various different infectious diseases, hepatitis B, hepatitis C, etc., and also various rheumatic diseases like rheumatoid arthritis, psoriatic arthritis, systemic lupus erythematosus, osteoarthritis, etc. To make sure that what we end up with is a specific list of protein biomarkers. And then we will select the combination which provides the best sensitivity and specificity. And this may end up with a combination of 10 biomarkers. We're looking for a particular, some of them may be raised in chronic fatigue syndrome, some of them may be decreased. So I'd like to end with a couple of slides which summarize the work of Robert Dancer, I think into which our work fits. And this is a hypothesis for the pathogenesis of chronic inflammatory disease. Here we have the immune system. This is the monocyte. These are virus protein subunits which have been engulfed by this cell and presented to the T lymphocyte. Cytokines are then produced as this cell becomes activated. Raised cytokines in the peripheral circulation may then affect cytokines in the brain through 3 different mechanisms: via the blood-brain endothelium, by activation of microglia, and by afferent nerves. Raised levels of brain cytokines may then produce abnormalities in the hypothalamic pituitary adrenal axis. And additionally, the efferent vagus activation attempts to decrease and control the immune responsiveness. These 2 aspects can together give the symptoms we know to be associated with chronic fatigue syndrome: fever, fatigue, pain, sleep, depression, and sickness behavior. And incidentally, these symptoms are not unique to chronic fatigue syndrome. They are what we all experience when we have an acute virus infection, and this is where this work from Dancer was initiated from. There is a biological basis too for the genesis of depression in chronic inflammatory disease, in that stress and chronic ongoing inflammatory disease can impact upon the immune response, levels of brain cytokines, increase indoleamine 2,3 dioxygenase, reducing levels of tryptophan, which are known to be associated with production of depression. So in summary then, we're making progress, and maybe in one more year, hopefully I'll be able to say we have published this and I can talk in detail about it. We'll maybe make some more progress towards development of a test. We certainly will make some progress in collaborations towards identifying the immune and virus-mediated pathogenesis of chronic fatigue syndrome. Thank you.